All right, guys, here we go at the start of chapter 12. This first image that you see shows the whole cell cycle and how we used a fluorescent dye to capture each one of the different stages. And so we're going to be going through this from start to finish. And a lot of this should be review. Now, keep in mind that it was Virchow in 1855 who came out and stated omnis cellula e cellula, that every cell is from a cell, so that all living things come from pre-existing cells. Now I just got to throw this in here, but there's some evidence that shows that Virchow actually stole this research from someone else, but he's still accredited with it. So I don't know, just thought I'd put it up there. So in this chapter we will learn how cells reproduce to form genetically equivalent daughter cells. So the whole purpose of this cell cycle is for us to form daughter cells that are identical to one another and that are identical to the parent cell. Now keep in mind, most of this chapter's content should have been in your Bio 1 class. This should be a review, therefore we're moving quick. So let's take a look at the rules of cell division. One rule is reproduction. Take, for example, a unicellular organism, like we see down here with our amoeba. It is dividing into two cells simply to create another individual organism. And by doing so, it will have the same genetic information as the parent cell. So we see reproduction for a unicellular organism. We see growth. Growth and development, especially for you guys, some of you are still going through this phase. Some of you are done, as in you're not going to grow anymore, but we still see development occurring. The picture below shows growth and development for a sand dollar. Okay, and the last role is renewal and repair, and this is something that all living organisms go through till the day that they die. They're done for. And so as you can see down here, we have some tissue renewal occurring in bone marrow cells to replace those cells that are just old and decrepit and broken down or worn out. So renewal and repair is extremely important. But in all cases, the cell division must distribute identical genetic material to two daughter cells. We want those daughter cells to be exactly the same as the original parent cell. And we'll talk about, well, what happens if that doesn't occur? And a lot of times that's a problem. So before we start getting into the cell cycle, I wanted to attack a couple of vocabulary terms. One being the genome. And the genome is the cell's hereditary endowment of DNA, otherwise known as the cell's genetic information. And this is usually packaged into chromosomes for manageability, especially when it comes to cell division. Now the genome, it's a massive amount of DNA. I mean, if you were to look at an individual eukaryotic cell, you're going to find about 2 meters of DNA. That's ridiculous that so much DNA is packed into such a tiny space, because we can only see cells with a microscope, and yet we got about 2 meters of DNA crammed in there. Chromosome structure. So we've often heard about chromosomes, okay? But what is a chromosome composed of? Well, chromosome is made up of a DNA and protein complex called chromatin. And those proteins are incredibly important. It is what the DNA wraps around. And there's this image, I believe, hold on, let me look. Oh, figure 4.8. It is titled Nucleus and its Envelope. And it shows you the protein there and how the DNA wraps around it. This helps to give structure and organization. So the proteins serve a purpose as one to help with the activation of genes, but they also serve that purpose of maintaining structure. It's kind of like if you take a ball of yarn and you wrap it around a core, so that way it doesn't get all tangled up and become a hot mess. Now, during cell division, this chromatin is going to become highly condensed into the chromosomes. So when we look at chromatin, it is this long, thin, linear piece of DNA. And right before cell division, it's going to coil up into that picture of chromosomes that we see, which looks like this. So here we can see our chromosomes coiling up, and that's what makes them visible. If we look at this picture down here, you can see the nucleolus right here. And within the nucleus, we just see the chromatin. It's not coiled up, it fills the nucleus, and it's still existing in that single strand. And this is where the majority of time is spent for a cell. This is how it does its functioning. This is what it looks like when going through replication. Only when we get to cell division do the chromosomes start to coil up and we start to see their appearance. So their structure, again, at cell division, each chromosome has already been duplicated. So after the replication of that DNA, we're going to go into our cell cycle. So the duplicated chromosome consists of two sister chromatids. Now these sister chromatids are identical to one another. They are attached by what we call a centromere. It is the point where the two sister chromatids are connected, and this is genetically determined by the sequence of the DNA. Now the chromosome number is going to vary in between species. Again, human somatic cell is a body cell. So it's every cell in your body except for your sex cells, which would be the egg and the sperm. But all other cells in the human body contain 46 chromosomes. In a cabbage plant, they have 18. Hedgehogs have 90. One species of algae has 148, which is ridiculous. So a long time ago, we used to think, oh, the chromosome number reflects the complexity of the organism. Obviously, we don't see that now as comparison of one species of algae, 148, and we only have 46. So in this image here, you can see I have my chromosome before replication. Okay, so this is an uncondensed, unduplicated chromatin. Okay, and what we see here is, so for at this stage we have our centromere right there, and that's what we can kind of consider as the waist of it, and it has two arms, one arm here and one arm coming off the other end. We then duplicate that DNA, and when it's duplicated, now we have two identical sister chromatids that are joined together at the centromere. And again, you can see right here is our little waist, okay, and they are identical to one another. Now, through the process of the cell cycle, we are going to get a separation of the sister chromatids so that each new daughter cell has a set of that genetic information. And remember, these are identical. So each of those daughter cells will have the exact same genetic information. So typically the goal of cell division can simply be stated as to split the sister chromatids and give one to each new cell. So that at the end, we have two identical cells. So let's start taking a look at the cell cycle. Parts of the cell cycle. The first part is what we refer to as interphase. 90% of the cycle occurs in interphase. So, and this is when the cell grows and duplicates chromosomes. The other 10%-ish, maybe not even that, is the mitotic phase, when the chromosomes are split into separate cells. So if we actually take a look at this, studies have shown if we were doing a cell cycle in 24 hours, that the mitotic phase right here would take about an hour. That's it. G1 phase is going to take about 5 to 6 hours. The S phase, or when DNA gets replicated, is going to take us about 10 to 12 hours. And G2 is going to take us about, I think it's like 4 to 5. Okay? So let's take a look at interphase, because that is where a cell is going to spend the majority of its time. Within interphase, there are three parts. There is the G1 phase, which we refer to as the first gap. And this is when the cell grows and carries out regular biochemical functions. So at this time, this whole process is the cell preparing to divide. It's just getting bigger. It's going through its normal everyday functions. Your liver cell is detoxifying, doing what it needs to do. Your heart cell's pumping. Your blood cells are carrying oxygen. This happens in interphase. In the S phase, S is for synthesis. So when the DNA is replicated or synthesized, and this is when we get the replication of chromosomes. Now, we're not going to go into great detail on this because we got a whole big chapter in order to cover DNA replication. Okay? And then finally we have the G2 phase, which is the second gap. And this is when the cell completes preparations for division, where it continues growing, where it doubles in the number of organelles. It's going to start, it's gearing up for reproducing. Now, it is of note that a cell can complete the S phase, but it can also fail to enter G2. And we'll talk about why in a little bit, meaning that it can complete that DNA replication, but then never enters the G2 phase. So again, taking a look at our picture here, this is all interphase. The majority of our time is spent here. We only see about 1 hour occurring in the mitotic phase. That is it. Not very much time is spent there at all. And again, a long period of growth, DNA synthesis, where we're going to replicate our DNA. And then we have the final stages of growth and where our organelles are doubling in number and so forth. Again, the majority of time that a cell exists is spent in interphase, and this is where it's going to do its everyday functions. All right, that's it.